Hi, and welcome to Bio 131 Lab. We're taking up the section on prokaryotes, focusing mostly on bacteria today. The section will be divided up into several parts. The first part today I'm going over will be cyanobacteria. Then we'll have a section on using the Gram stain and looking at different types of procedures for identifying shapes and colors in the response to the stain of the bacteria. Then we're going to have a separate section that will be on antibiotics and testing them and how we can read that in a plates comparing a gram positive and a gram negative, and you'll make a graph. And then the last part, we'll be doing a field trip to a water treatment plant because a lot of what's going on in treating wastewater has to do with bacterial prokaryote properties. And in that case, we'll talk about the archaea, the separate domain in the prokaryotes. And so right now in this video, I'm going over the cyanobacteria. So let's get started with that. So cyanobacteria, we're going to go over a couple things. I want you to learn about the characteristics of the cyanobacteria, what makes it a cyanobacteria. Cyano, to begin with, is blue-green color, and that's why they're sometimes called the blue-green algae. Well, we're going to talk about the significance of nitrogen fixation. It's one of these biogeochemical cycles, and I'll go over what that term means. Then we're going to learn to identify three different species genera of Cyanobacteria, Anabaena, Gleocapsa, and Oscillatoria, each of which I've chosen to show you some unique features that represents the other parts of the Cyanobacteria domain or kingdom or phylum that we aren't going to be seeing. Okay, so let's get going on this. And so first, characteristic of Cyanobacteria. You can see they're in the domain bacteria. I have circled in purple down there, so you can see they're its own separate group. So bacteria's domain within it, these would be that same concept we'll talk about when we get to the protist supergroups. I'm going to use cyanobacteria as meaning a kingdom. So we'll just call it the cyanobacteria kingdom. Technically, it could be a phylum depending on how you're breaking it up. But for us, we'll just call it a kingdom. Just remember they're cyanobacteria. And their color is blue-green, and that's what gives them their name. They're really important in what we call biogeochemical cycles. And so the bio means life, obviously. Geo is because it's soil-based, so it's something going on in the soil. And a cycle because it transforms from one chemical form to another chemical form to another chemical form to another chemical form and ends up back where it started. So it's cycling around. And so the importance of nitrogen in the world is in biology is particularly because you need nitrogen to make amino acids. It's the amino group, the NH3, and reduced, but anyway, the NH part of the amino group and the amino acid, and then that forms proteins. And proteins are important because they're structural, like our muscles, but even plants that don't have a lot of protein content, a lot of them mostly, need protein because proteins make up enzymes, and enzymes run their metabolism. So any living organism with metabolism needs protein to make the enzymes, and to do that, it needs nitrogen. And so most organisms that aren't bacteria are getting, animal-based, let's put it that way. You get your protein in your diet from eating other things made of protein. So you eat, let's say if you're not vegetarian, you eat a hamburger. It's got a cow. Where did the cow get it? Well, the cow ate the grass. Right? And so it incorporated it into its body from taking it in as protein. Plants make protein. And so they make it from scratch. They don't eat protein from the soil. They take in nitrogen. And so looking at that, how nitrogen gets into the soil is really important. And I'm just going to focus in on this lower spot right here on the left-hand side I put in purple. And that is nitrogen fixation. Obviously, nitrogen cycles, so as plants die, they decompose. Animals die, they decompose. Their waste products, their urine, their poop, that all contains nitrogen, and that decomposition is an important part in the soil as well. But bringing nitrogen in, the biggest pool is in the atmosphere. And so we're going to see that nitrogen from the atmosphere, NO2, can be brought into NH3, into the soil, that will get incorporated into plants, and then that can start feeding the cycle. And so nitrogen fixation is really important. I bring that up because a whole group of cyanobacteria or organisms that can do it. They're not the only ones. There are other bacteria that can do that aren't photosynthetic. But cyanobacteria are some of the most independent organisms for doing photosynthesis in that they just need water, sunlight, they need some micronutrients, but the main nutrient that they need that you put for plants in fertilizer, nitrogen, they can get from the air. So they could just grow. And so they're a very significant factor. Other thing about, I guess this is not fitting on the screen a little bit, so I can make it a little bit bigger. That's a little bit off. Sorry about that. But anyway, one other thing that you'll hear about cyanobacteria is around here in the summer, they have this tremendous, what we call blooms. And so they grow in unbelievable numbers, and that causes some of them to become toxic. They can be toxic to us if you're swimming in the water and you take in more. We usually don't drink a lot of water when we're swimming, but dogs do. They take in quite a bit. And so you'll see posted around lakes and things around here, or there'll be warnings not to let dogs go into the water at all. They can contaminate drinking water if there's a high enough bloom concentration. Usually, of course, the drinking water has been purified, but it makes it a very expensive process, so they get the toxins out, but often it leaves a very odd flavor to it, or odors to the water. And so they're a big problem. So here's just showing you some of these harmful algal blooms that we have. Just showing you some on the Klamath River. These are just, I'm just pulling this up on the internet really quickly right now, and these are from November, right, and December, talking about what happened last summer and how they're planning on taking care of that in the future, right? And it's a big problem, and so it causes, when we get to talk about the water treatment plant, we'll have the same problem, what's called BOD, biological oxygen demand. And when these algae grow really fast, when they die, they sink to the bottom, and then other organisms decompose them and they use up the oxygen. So it actually can make the water, it becomes toxic, so it actually can just kill the fish that's in the water or other organisms, especially marine mammals sometimes. And also it can make it so the water has too little oxygen for fish and things to survive in it. So they're a significant economic issue, an ecological one. This is Lake Chabot. When I was hiking there in the summer, I could see the warning there telling you to watch out and not let your dog drink the water. And you can see how green, that's that blue-green color. You can see some of it looks similar to plants, but there's also a little bluish color to it. And we'll talk a little bit about that when we get to the pigments. Okay, so the first one we're going to learn about is called Anabena. So we have three. You want to pay attention to how would you identify these on a lab exam. They look very different, so I've chosen three. They're easy to tell apart. And each one has features, right? So the most complex one we have is Anabena, and this is probably one of the more complex. So this would be looking at your slide, if you're in lab, it's not the best focus here, on 4x, right? So low power, actually it'd be more like 10x. But we take it up to oil on the slide, and under oil it would look like this. And I mentioned this is the most complex because usually bacteria are a culture of all the cells are the same. But in this case, you're going to see that there are three different sizes and shapes of cells doing three different things. The vast majority of all the cells are all this blue-green. This has been stained. If this were a fresh material, what is pink here would have been a clearer color, slightly less green. So, and I'll have a picture of I'll zoom down to it. So in our fresh material, you'd see this one is clearer. One of these is a little bit clearer. Then you have one that's much larger, and then you have all the others that are pretty much the same, right? So those are our three different types of cells. So what are they? And so what they do is we have ones that are called heterocyst, and the name literally means other cell. And that's the cell that looks different from the others, and that's the one that does nitrogen fixation. So it's providing nitrogen to the rest of the colony. The way this grows is one on top of each other in a long chain, and that's called a filament. So this is a filamentous bacteria. More specifically, if you've taken micro, this would be a trichome, but we just call it a filament. Right? And so you can see the heterocyst here. It's clear. This is not stained. You can see it's got two little dimples on either side. That's a little indentation of the membrane that's probably important for an invagination of the membrane for doing this nitrogen fixation. Then you have what we call vegetative or somatic cell. Soma simply means body. So somatic cell is a body cell, right? We don't, quote, have body cells because in our body, either it's a muscle or a skin or nervous cell. But in a lot of organisms like plants, There's a whole bunch of cells that are all the same that kind of fill up most of the space. So we refer to them as somatic or body cells. Right? They're the bulk of it. And then we have a large cell, much larger than the other ones. And so the small cells are the somatic cells. Just slightly larger and clearer will be the heterocyst, or stained pink in our slide here. So those will be the heterocysts, pink. And then what you'll have is a cell that's about the size of 3 somatic cells. It's much larger. And this is still young, but when it's more mature, it will have a very thick cell wall. And this is a type of spore called an aconite. And what it is I call it a resistant cell. So these, in the spring, when it's raining around here, you'll see this, what we call pond scum, this kind of blue-green stuff growing on the surface and even in the wet soil next to it. That's cyanobacteria. But what happens in the summer is it all dries up, and the somatic cells die, the heterocysts die, but the aconites, that's the resistant, they can survive either freezing, if it freezes up in the winter up in the mountains. The aconites survive to start growing when it melts. Around here, if they make it through, our strict period is the long, hot, dry summer. And so they're making it through that. So that would be the aconite, or it's a type of spore, resistant cell. Okay, so you can tell this is Anabaena because there's these 3 different types of cells. They're kind of roundish, and they're in a long chain called a filament. We will see another filamentous Cyanobacteria oscillatoria, but the cells aren't round, right? They're like little disc shapes, and they all line up, so you can't even barely tell one cell from another. So they look very different when we see it. Okay, so this is Anabaena. And if you have your lab manual on page 21, we're starting, so you want to label those 3 different types of cells on your diagram there. The next organism, and I have some more pictures of Anabaena, so you can see the heterocysts and the somatic cells and the aconites. So take a minute, see if you can see, there's only one aconite here, see the difference in them. Then I have them labeled there for you. Another view, so take a look. And so here, those are our heterocysts. In this prepared side, they're staining pink. Then we have the much larger aconites. And then all the other cells would just be the somatic cells. So those are 3 types. And so that's our Anabaena. Really common organism. So if you go out to ponds around here, you can pull up a lot of this stuff, right? It makes kind of like long threads. And so if we're in lab and seeing it in a test tube, it'd be settled down at the bottom and then kind of like in a dark, because when they ship, they kind of all coalesce together. And then you put your pipette in and you stir it up, and then they spread out. So the whole thing looks like it's bright green water, but when you put it on the slide, that bright green is really little strings in the water. So it's kind of neat to do that when we see the fresh stuff. This is the fresh one that I already labeled for you. So you see the heterocysts here are the ones that are lighter green. That's another heterocyst. You can also say that they're just about slightly like 1.5 sizes of the somatic cell. Aconites are more like 3 times as big. And notice they're not clear. You see a lot of cytoplasm in them. They become very densely packed with cytoplasm. They have to change their cytoplasm as well as the cell wall material in order to survive either freezing or drying out. So they're doing something that will protect them. They're not actively growing. Okay, the next one I took up is Gleocapsa. And the reason I took up Gleocapsa, when you see it, it looks like a green dot, just a little green dot. But what it has is a feature that we've talked about for bacteria, and that is a capsule. And that's that gelatinous, proteinaceous covering to the outside that they can use for protection and to adhere onto substrates. And here it actually gives them a little protection, and it kind of gives them, they kind of stick together. But the neat thing about this one, Gleocapsa, is so thick you actually can see the different layers. As it grows, it continues to make this capsule. So here I have a diagram. You can actually see an individual cell, and there's its capsule being made to the outside. Then this one divided, right? This, and then it would have the original capsule to the outside, and then the daughter cells would make capsules to the inside, and then they divide, and this is making one on the inside. So you can get these layers as they divide and spread out and divide and spread out. And so here, here's an internet picture of Gleocapsa I got. You can see the outer covering of the original capsule here. We'll draw it in. So here's the original capsule. And then it's dividing here. We'll make another capsule to the inside. This is a slide where they actually used a very fine powder on it, kind of like ink. What it did is it stuck to the capsule. So this one has been stained in a very unusual way. So you can see that what would be dark here, those would be the actual individual green cells. Another thing I want you to notice about cyanobacteria, so here's a fresh material from one of our labs earlier on. And so you can see one level, that was the capsule. Then it divided, and now the new daughter cells are making the capsule on the inside. And they'll continue to do this, and actually you can see when it was one single cell, that was the capsule out here. So you actually can see as it's divided into the 4 cells, first it was one, the capsule to the outside, then that capsule enlarged as it became 2 with their capsules, and then that divided, and each one of these became the 4. And so here's another view. This is a live material that I just used the microscope in the lab and took a picture of. You have to fiddle around when we're doing this in lab and learning about the microscope. For this, is a good opportunity to play with the iris diaphragm and the iris diaphragm and the dimmer switch, because you need to turn down the iris diaphragm all the way to see these clear areas to the outside. And so all students there, you open the iris diaphragm, you can't see anything with little green dots. You shut it all the way down, and then that makes it dim, so you got to open, add a little bit more light. And then you can begin to see the capsule. So remember, when we're looking at very clear things, this is when we'll be playing with the iris diaphragm. So if we were live in lab, it would take some time to go over that. So anyway, here you can see, obviously, the bright green are all the cells. And I would ask, how do you know this is bacterial and not plant cells? So we saw Eludia in the very first cell lab learning the microscope, and we saw little green dots of chloroplasts. And if we did our measurements, you'd see that these cyanobacteria, especially these Gleocapsa, are about the same size as the chloroplasts. Right? And where in the plant cell you had one large cell with little green discs on the inside, in the cyanobacteria they do not have chloroplasts. The whole cell membrane is doing photosynthesis. So the whole cell is green. There isn't like a subset of the cell doing photosynthesis. The whole cell is doing photosynthesis. So this is our Gleocapsa, right? Very different looking from Anabaena. You won't mix it up for that at all. Okay, the last one we're going to look at is Oscillatoria. And by a name like that, you may wonder, why is it called Oscillatoria? Well, it oscillates and it does something that's called gliding motion. And so you can see it is filamentous like Anabaena, but unlike Anabaena, they're not rounded cells, they're more discoid or disc-shaped, where they stack right on top of each other. And so you can see the little lines in between. What you see, those little dark areas, those are cells that died or got a little air bubble or something in the preparation of the slide, because this is a prepared slide. And air bubbles under the prepared slides just leave little dark areas. The way this grows is just divides end to end, so it's a bacteria, so it divides by binary fission, just like Gleocapsa was doing. I was showing you how they stick together in the capsule. Here they just divide end to end, and they're all dividing, so the filament gets longer and longer, and finally it breaks, and they have gliding motion, so they'll actually slide away from each other. And I'll show you a little video of that in a minute. So this is Oscillatoria. And so it has a gelatinous sheath to the outside. It's thinner than Gleocapsa, so we would not be able to see it in lab, even under our oil lens. And that's why I chose Gleocapsa, or I use it, because its capsule is so thick you can see it easily in the lab with a light microscope. Other ones, the capsule is not nearly that thick. Okay, here's another view of our prepared slide with Oscillatoria, where you see this area and it's swollen and it looks kind of football shaped. That's an area where the cells are actively dying and pushing apart, and that's where it will break and then slide apart, right? And so that looks a little bit different from just little dead cells that I circled here. That's probably just an artifact, we call it, something that went wrong, or something that changed during the preparation of the slide. It has nothing to do with the organism. We call that an artifact, right? Here, we use that is actually growing in the organism, and that is a breaking point. It will actually break off. And if you actually look at the ends of this one, you can see how the tips are kind of cup-shaped. And that's because that's what's happening there. It's making a little cup, a little air bubble as they die off. And you see another one forming down here. So that's what's going on in that slide. So Oscillatoria is another very common, and you can see how the tips kind of swell up. That's after it's broken. It's a very common water algae that we have growing up in our ponds mixed in. So, if you were to pull this out of a pond, you'd get Gliocapsa, you'd get Oscillatoria, Anabaena, Nostoc, a whole bunch of other ones. It's not like a pure culture, but these are some of the more common Cyanobacteria that we'd find in our ponds. And if we're in lab, we just go out to the pond next to the ATC building and pull up some of the green stuff and take a look at it. Right? Down in the music building, the ponds were so contaminated with the cyanobacteria. It was causing problems with the ducks that were down there, and you occasionally even have a river otter in there and thing going after the crayfish that live down there. So you'll notice when you're on campus that in the summer, late spring to summer, they put in this blue dye. And what the blue dye does is it absorbs a lot of the sunlight, so it makes it hard for the algae to grow, and that keeps them from getting to the bloom levels, right? Obviously, the dye is safe for the fish and everything. You can actually get it for ponds at home. They sell it in different stores. But anyway, that's why that pond has that weird color in the summer. They do that particularly to keep down the cyanobacteria. Okay, and so what I'd like to do is I'm going to shift over and have a quick little video. And showing this gliding motion. And so they're moving. This is Oscillatoria. You can see some of the clear areas there. That's also an artifact of them making this thing, but they're alive. And this is, from what I've seen in lab, I don't know for sure the background on this, but this is sped up quite a bit because in lab they do not move this fast when we see them. But they do move, right? So if we're fresh in lab, we'd actually see this going on. It's a gliding motion. And gliding is one where you see movement, but there's no appendage. There's no flagellum. There's nothing sticking out to the outside to figure out how it's doing it. I've read, and the best thing they can think of, you can see how it kind of twists a little bit as it's moving. They have microfibrils in there, and somehow they think it's actually pulling and actually gliding in its gelatinous sheath to the outside. So this is another type of movement, not with flagella, very different, called gliding movement. We'll see another organism later on, diatoms, also do a gliding movement when we get to the protists and the eukaryotes. Okay, so this concludes the cyanobacteria. The next video you want to watch is beyond how to do the gram stain, and we'll be doing that next.